Hey everybody, welcome back. Let's take a look at Unit 6, Lesson 2, Exploration 1. This is the second lesson in Unit 6, and where we looked at the structure and function of DNA in Lesson 1. In Lesson 2, we're going to focus on this idea of what's called protein synthesis. How do we actually use the information that's found in DNA to create, in this case, proteins? And so that starts with this idea of protein synthesis. Now, in the next two explorations, we'll look at the two stages of protein synthesis. But in this first exploration, it's just kind of an overview of both of those and kind of how we came to discover it. So let's start out with something called the central dogma. When Francis and Crick discovered the structure of DNA, like we said, along with some help from other scientists like Rosalind Franklin and Erwin Chargaff, they developed what they called the central dogma, which basically says that the information found in DNA is used to create proteins. And that flow of information does not go the other way. Proteins aren't used to create DNA, only in one direction. And again, it's kind of like a cookbook being used to create some sort of meal. The instructions are found in DNA to create the actual physical product, which is those proteins. So I want to look at a couple key terms that you're going to want to be familiar with. We've used this word gene before, but again, a gene is a specific piece of DNA that provides instructions for how to make a specific protein. You know, your textbook says that humans have about 19,000 protein coding genes. Another way to think about that is like 19,000 different recipes for specific meals that your body can create. When your body needs something, it makes a protein. And so there are approximately 19,000 different proteins that your body needs to be able to make. And so you have that many different genes. Now, there are more genes than that in your DNA, but some of those genes, we don't necessarily know what they're used for, if anything at all. They might just be what we call junk DNA, but we might talk about that later. Let's see. When we talk about the connection between a person's DNA and what they actually look like, the proteins are what kind of connects those two things together. Your body creates proteins that make your eyes a certain color. It creates proteins that make your ears a certain shape. Every aspect of your physical body that you inherit from your parents comes from your DNA and is produced by specific proteins that allow those structures to come together. All right, so let's take a look at some of these different examples of types of proteins. This table right here shows some of the major functions. And we're not going to go through them all necessarily super directly, but I want to highlight a couple things. First of all, storage. Proteins are very big in the storage game. They help your body transport things like we looked at in a lot of different areas, but also in the cell membrane. Proteins are huge in maintaining homeostasis. They are the chemical messengers. They also serve as antibodies to help your immune system function. They provide the two major chemicals for muscle function. When your muscles fire, they have myosin and kinesin, basically allowing serious amounts of electrical energy to be passed and then transferred into that contraction or release. They're a part of the structural element of your body, both in your bones and in your finer tissues like skin and hair. And as we talked about earlier, they can serve as enzymes to make reactions happen faster and easier by lowering that activation energy. So, here are some examples of those enzymes. Lactase is a really important enzyme that helps your body process lactose, which is found in a lot of dairy products. All right, so proteins have a lot of different functions. They're very important. Let's kind of talk about how this process takes place. Now, again, there are two stages of protein synthesis. They're called transcription and translation. And again, we're going to go into much greater detail about each of these two stages in our next two explorations. But Let's briefly introduce them here. Transcription, think of transcribing. If you are transcribing something, then you are making a copy of it. Transcription is where we take a sequence of DNA and we create a copy of it on something called RNA. Now, we haven't talked about RNA yet, but we will in a second. RNA, and in this case specifically messenger RNA, is a disposable copy of DNA. Think about it like a Post-it note. If you were gonna write something down and it just needed to be a temporary information storage, you have a pass to another class, or maybe you just want to write down a quick, code or password or something like that where it doesn't need to last forever. We're not going to put it in a book. It doesn't need to be able to, be durable or anything like that. It just needs to carry information for a small period of time, maybe from one place to the next. That's what messenger RNA is. A very temporary, very easy to make, but extremely disposable molecule that is able to carry a copy of a DNA message to where it's going to be put together. All right, we wouldn't want to take out the actual piece of DNA because then once you remove it, you can't use it again. So messenger RNA allows us to make a copy of a section of DNA and then immediately dispose of it because it's easily recycled. Here's a diagram that if I was you, I would also fill out the way I filled out. This kind of shows you that flow of information, that central dogma, the idea where you have DNA that's copied into messenger RNA, and that process is called transcription. And then the next process, which we'll talk about in a second, is where you take that messenger RNA with that Post-it note style of information, and you translate it into protein. So we have transcription and translation. A couple key things. First of all, transcription, as you can see here, takes place inside the nucleus because that's where the DNA is located. Translation is going to happen in the cytoplasm, which remember is that area outside the nucleus, but still inside the cell. And that's where we're going to make our proteins. All right. Like we talked about, transcription takes place in the nucleus. Translation occurs in the cytoplasm. Make sure you have that noted down. And then one more thing, and this we'll probably talk about a little bit more in our next exploration, but an important aspect of translation that happens is called mRNA processing. We are able to essentially take that mRNA and only include the essential pieces. And again, we'll talk about why that's important in our next exploration. All right. We mentioned RNA. Now, RNA sounds a lot like DNA. It's a nucleic acid. It's in the nucleic acid family. There are a lot of similarities, but there are also some key differences. Like I highlighted here, RNA is like a temporary copy of DNA. It's easily formed, but then it's easily recycled. Think a Post-it note compared to a textbook. DNA would be like the textbook of information. Hardback cover, heavy binding, very durable, meant to last for many years. RNA is like a Post-it note. Very flimsy, very easy to create, but very easy to throw away. Not going to be very durable, but it serves a different purpose. And so that's kind of like the difference between DNA and RNA. DNA is a much more permanent molecule. RNA is very temporary when it comes to temporary information storage. So let's look at a couple key things. First of all, let's compare some of the similarities. Both DNA and RNA are going to include cytosine, guanine, and adenine in their nitrogen bases. Both of them are going to use those 3 nitrogen base pairs. But take a look down here. In DNA, we use the nitrogen base thiamine, which is not found in RNA. Instead, in RNA, we'll use uracil. This is going to be important when we talk about how the mRNA molecule is actually created, because what we're going to do is we're going to pair up with the complementary strand. But remember, when you're creating RNA, it's going to be important that you remember to use a U, because thiamine doesn't exist in RNA. So that's one key difference. The second one you can see just by the picture. DNA is double-stranded. RNA is single-stranded. And again, that goes back to kind of that structure, that durability, where DNA needs to be extremely durable, extremely sturdy. RNA doesn't. So it can be single-stranded and not closed. There's an exercise that I would highly recommend that you guys go through right here. where we can compare and contrast DNA and RNA. Now, I've already finished it for you, so if you want to pause the video and then go back, go back and match it up. But I'd encourage you to try to do it on your own and then just see how it goes. But here are the similarities and differences of DNA and RNA. They're both nucleic acids. They both have that sugar phosphate backbone. We mentioned that they both have adenine, guanine, and cytosine, but key difference, DNA has thiamine, RNA has uracil. DNA uses a deoxyribose, RNA uses a ribose, which allows DNA to be double-stranded. RNA is single-stranded. All right, so there's your introduction to protein synthesis. We'll check back in later with the next two pieces of this. But for now, that's where we'll finish.